Long COVID. Underlying mechanisms. Long COVID likely reflects multiple overlapping biological mechanisms that vary across individuals. Viral persistence is theorized because SARS-CoV-2 RNA and protein fragments have been identified in tissue months after infection, raising concern that ongoing viral immune interactions, similar to mechanisms observed in HIV/AIDS and EBV-associated multiple sclerosis and lupus, may drive chronic inflammation. Immune dysregulation is proposed because persistent inflammatory signaling, abnormal cytokine profiles, and disrupted T and B cell function have been observed months after infection. Some patients demonstrate diminished or atypical antibody responses following vaccination, impaired immune memory formation, and reduced responsiveness to subsequent infections, suggesting sustained immune imbalance rather than full immune recovery. Autoimmunity is suspected due to the detection of autoantibodies and immune responses targeting host tissues following SARS-CoV-2 infection. This may help explain new-onset autoimmune diagnoses and autoimmune-like syndromes observed in some patients, such as lupus, IgG4 disease, Sjogren's syndrome, and rheumatoid arthritis, amongst others. Latent viral reactivation is considered because immune instability can permit dormant viruses, including Epstein-Barr virus, cytomegalovirus, herpes simplex virus, among others, to reactivate. Reactivation may further amplify systemic inflammation, immune activation, and overall symptom burden. Endothelial and microvascular dysfunction are theorized because SARS-CoV-2 can damage the lining of blood vessels and disrupt normal vascular signaling. This may impair oxygen delivery, promote clotting abnormalities, and contribute to multi-organ symptoms. Autonomic dysfunction is proposed because SARS-CoV-2 may impair vagus nerve signaling and its bidirectional regulatory control over immune activity, vascular endothelium, baroreceptor reflexes, and autonomic ganglia. Disruption of these interconnected neuroimmune vascular pathways can destabilize heart rate, blood pressure regulation, vascular tone, and inflammatory modulation, contributing to systemic physiologic instability. Fibrin amyloid microclots are hypothesized because studies have identified structurally abnormal, amyloid-like fibrin aggregates that appear resistant to normal fibrinolysis. These persistent microthrombotic structures may obstruct capillary circulation, impair microvascular perfusion, and reduce tissue oxygen delivery. Neuroinflammation is suspected because evidence suggests sustained immune activation within the central nervous system following infection, including microglial activation, astrocyte reactivity, altered cerebral blood flow, and blood-brain barrier disruption. These patterns parallel mechanisms observed in other neuroinflammatory conditions, such as multiple sclerosis, while remaining distinct in underlying causation being COVID-19 infection. Microbiome disruption is theorized because reduced microbial diversity and altered metabolite production have been observed following SARS-CoV-2 infection. These changes may impair gut barrier integrity, promote microbial translocation, and disrupt immune and metabolic signaling via the gut-brain axis, contributing to persistent systemic inflammation and multisystem symptoms. Long COVID is likely multifactorial, with overlapping and interacting biological mechanisms rather than a single unifying pathway. As the science evolves, maintaining intellectual humility and openness to emerging evidence is essential. Research frameworks and care models must remain adaptable, multidisciplinary, and responsive to new discoveries. Rapid, coordinated dissemination of findings, through public education, clinician training, and health system integration, is critical to translating mechanisms into meaningful prevention, diagnosis, and treatment strategies. We are all one infection away. Learn more about long COVID at longhauler-advocacy.org.